Final two speakers. Are you ready for some more honest, true stuff of what's really going on? Our next speaker is my good friend, Mark Davis, who is the Avery Professor of Immunology at Stanford. He's the director of the Stanford Institute for Immunology, Transplantation, and Infectious Disease, and his current research is focused on human immunology and understanding the immune response to cancer, vaccination, and infection. The title of his talk is Immune Organoids Open Up a New Window into Human Vaccine Response and Dysfunction. Thank you.
Well, I'd like to thank the organizers, except I am one, so that's probably not completely appropriate. But thank you. And yeah, it's been fantastic. Thanks, Lucy, for making this happen, really. She's responsible. And it's so really been great. I've learned a lot from this already. So, and maybe I have something to teach people. And I think one thing in terms of, we've heard a lot of different aspects. This is like the blind man and the elephant story. But here's my take, which is that whereas the SARS-CoV-2 vaccines have been incredibly effective, we were lucky. There's a lot of vaccines that have failed. And there's this really long list, actually, of some very major pathogens that we have no idea how to make vaccines for. We keep trying and fail. And then if they fail, we actually don't even know why they fail. Clearly, if you do what Pasteur did or you do variations of that, you can cure a lot of disease. We have, measles vaccine, we have yellow fever. Vaccines can be terrific, and they could just solve the problem. But we've been struggling with these other diseases for decades now, and again, we keep doing making new vaccines, and at great expense doing human trials. It costs about $500 million to do a human trial of a novel vaccine. And they go on for years, and then they fail, and again we can't learn from it because we have very little information. We can't slice and dice people after they failed the vaccine, so we can't really go into any depth. So it seems like we're doomed here, and one reason we're doomed is that the inbred mouse immune system is not the same as the human immune system. There are signs everywhere that it's different. There are hundreds, if not thousands, of mouse models of disease that provide interesting data that you get papers in top journals, you get grants from NIH, but then they don't translate into people. Some do, but actually, the vast majority don't. And they're just in different units. Now, why is that? Well, they're different species. They lack genetic heterogeneity. They lack microbial disease experience. They have unique genes and patterns. But I think the biggest reason, in my mind, is that you don't need a sophisticated immune system for inbred mouse. If you ask Mr. Google how long do wild mice live, you get the answer is about 6 months. So if you can keep a mouse alive for 6 months, that's great. You're going to have lots of mice. But if you can't keep a human child on average surviving for 20, 30 years, you're done as a species. So you need a lot. I think you need a much more sophisticated, capable immune system for human beings or long-lived primates than you do for mice. So that's one of my justifications for doing humans is I think there are going to be a lot of interesting new mechanisms of the immune system. And we don't know about them because it's been so difficult to study. And so we spent 20 years now developing new tools and new approaches to really look at human immunity in great depth. But one of these tools that I'm most excited about is a few years ago we found that we could take tonsils that are being ripped out of innocent little children mostly, and we could chop them up, and we could basically recreate a vaccine response in vitro. Not every vaccine response, but flu and others worked really well, and this is pioneering work of Lisa Wagger, who's now on the faculty at UC Irvine. But in this very simple method, you make single-cell suspensions, you let the cells reassemble, which is very different than most organoids. They will form germinal centers. They will produce antibodies, high-affinity antibodies. They will undergo affinity maturation. So that's a phenomena that only occurs in lymphoid organs where you stimulate an immune response and B cells are triggered that have the specificity are triggered to go into germinal centers, acquire mutations, and the ones that have mutations that increase the affinity of the antibody get to go forward. They also require T cell help. Anyway, this is an experiment where you can stain high affinity B cells in tonsil cells. You can eliminate them also, so you get rid of all the high affinity B cells with a staining reagent, and then you stimulate in the organoid culture with a flu vaccine, and you can get high affinity B cells back. That is, B cells that were there but low affinity are now being mutated and selected in germinal centers to be high affinity. So this is something you can't do with blood cells. So it really puts us on a whole other level in terms of being able to understand an immune response and being able to characterize vaccines. So what have we done with this? And I think one of the more spectacular things that we've done recently has been addressing the age-old issue of sin. And of course, that you say that, everyone's interested in sin, and people have been trying to do something about sin for, thousands of years, I think. But this is actually a smaller subset of the whole range you could imagine of sin, which is original angenic sin, which has been mentioned by Kanta and others. And so what does this mean? This is really it in a nutshell. If we stimulate a tonsil organoid from some random person, adult or child, with different flu strains, hemagglutinins, you can see a bias. In 70% of the people, you will see a bias. So here, this is a standard flu vaccine, has 4 different strains, including as listed here. But if you look at the antibody response, it really, you're only getting a good response to the H3 strain and pretty crappy antibody responses to the others. So this is the phenomena of strain bias. And as I say, most people have some bias of this sort or the other. And people have wondered for decades what is the cause of this. And that's where this doctrine of original angenic sin came about in 1960, and it was even thought about earlier. And the idea here is that the first flu infection you ever got biases your immune system towards that strain. And for long after, maybe forever after, that's what you're going to do. So even you get a vaccine with 4 different strains, if it has your favorite strain, that's what you'll make the most response to. But then if you get infected with another strain, you're out of luck, or you're not going to do as well. So that's why the flu vaccine has never achieved the efficacy of yellow fever or measles vaccine and so forth, because of this strain bias, and people are different. And this was, I don't think not without controversy, but it's been sort of the major dogma in the field since 1960. And also had a really attractive title, anything you could label as sinful, people gravitate, I don't know why. But VAMC who is featured in this this publicity photo that he has circulated, challenged this model. And he's a real flu guy. He did flu as a graduate student, and he really knows this stuff. I don't. And so we had historically had assembled a monozygotic twin cohort that we vaccinated with the flu vaccine one time, and not surprisingly, twin pairs, each member of a twin pair would have the same bias. But, so that could be genetic, but it could also be environmental because twins grow up together. We looked for twins that were separated at birth, not a one. There was nobody is separated at birth anymore, I guess, on twins. So that's very sad. But Vamsi dug into this and not only showed that the twins share biases, but that they don't share historical cross-reactivity to other strains. They're all over the place in terms of other strains. So the original antigenic sin model didn't really quite fit this data. Further, even more dramatically, in a collaboration with Mary Stott at Cincinnati Children's Hospital, she has incredible birth cohorts analyzed for flu. So every week since birth, the moms swab the noses of their infants and then send the swabs in to the hospital to be typed. So you know exactly when a child was infected with flu or that they weren't infected. And so we got 15 kids that had never had a flu infection, but were then vaccinated at 6 or 12 months, as you can be. And they exhibited a strain bias. 70% of them exhibited a particular strain bias. They had never been infected with flu. So this was also another, I think, nail in the coffin of the original SIN model. So then going back to the twin cohort, you're saying, well, it's genetic, but it's not original SIN. So what is it? And here you have to learn a little bit of immunology. I know this can be tough for some people, but how an antibody response happens is not a trivial process. It's basically a B cell with a particular surface antibody that might be specific for a flu antigen, like hemagglutinin, has to bind that and internalize that antigen, grind it up into peptides, and then it presents those peptides, as it says, bound to histocompatibility molecules, class 2 histocompatibility molecules. And these are what T cells see. So in the diversity of T cells, there'll be T cells that will see a particular flu peptide bound to a particular MHC. And if that happens in this sort of complex, it will then make cytokines that stimulate this B cell and allow that B cell to mature into an antibody-secreting cell, enable it to go into the germinal center and increase its affinity if that's what you need to do. But anyway, this is an important part of the cycle. And what you have to know is that there's an incredible diversity of class 2 MHC molecules. They are not all the same, and there are thousands of alleles. And so the next brilliant thing Vamsi did is he went to the immune epitope database, which you heard about briefly from Joe just in the last talk. And the nice thing with this resource is you can plug in any protein sequence and ask the program how many peptides would be presented by a particular MHC molecule. And so what you can see here is that if you look at the main strains, H1, H3, or B, these MHC class 2 alleles really like the H3. They make a lot of peptides for H3, but fewer peptides for B and somewhere in the middle for H1. And yet you can go down here to these other class 2 alleles, and they really like B strain and hate A3 strain in terms of peptides. So they're going to, there's a broad diversity from these alleles in terms of how many peptides they'll make from a given strain. And working with peptides that are predicted, Vamsi also showed that this is consistent in terms of the T cell help that could be provided, that they did follow this prediction. This is really just a prediction, right? So, but the killer experiment is this one, with using the tonsil organoids. VMC made a multimer constructs which had the hemagglutinin from the different strains separate on different constructs, just like the vaccine. The vaccine, multi-strain vaccines are all made the same way. You grow up the different strains separately, and then you mix them together in the final assembly. So this is a mock-up of the standard flu vaccine, but then the different thing is that you can also make a mixture. Basically put all these major hemagglutinins onto one construct. And what does that do? Well, if T cell help is limiting, which it normally is, what this will do is it'll bind to every B cell that is specific for any hemagglutinin from any strain. And so even the least favored B cells, the ones that are going to be the lowest frequency, giving the worst response, will be able to internalize and then present peptides from the more popular T cells. So it's a way of borrowing T cell help from the favored strains, the biased strains, for the least favored ones. And does that help? And the answer is, of course, that it does. This is, again, what I showed you before, a strain bias in this particular tonsil. The cocktail with the separate hemagglutinins in the mock-up gives you basically the same thing. But now if you, with this mixed cross-linked construct, now you see the antibody responses are now get very much more equal. And that's what you want. That's could really make a huge difference in how we formulate not just the flu vaccine, but other vaccines that you could then predict, which are like with RBD domain, we definitely predict there'll be biases for hemagglutinin. And then it has come up multiple times for good reason that what's really scary is a potential bird flu pandemic, H5N1, and you might say, well, I've had a lot of flu vaccines and infections, so I probably have cross-reactivity to avian hemagglutinin. And you'd be wrong, because here we've immunized with the avian hemagglutinin, and it's barely above background in terms of the antibody response in the tonsil system. But if you make a construct that is targeted towards those rare avian flu B cells, but you bring along all this T cell help from the other strains, now you can get a very decent antibody response. So this, and this isn't just about flu. It means you could borrow T cell help from flu to boost other responses. So say Elon brings a virus back from Mars, and you will have some B cells that will react with that, but they're not going to have a lot of help, and so they're not going to make a lot of antibodies. So why not load them up with these flu strain proteins that will provide all this help? So that's a good thing. We have also used these organoids in another way, which is to ask questions about regulatory T cells. So we know that T cells, that there are specific regulatory T cells, and they really, if they're not working, you're going to get autoimmunity and could be really fatal. And the best known one is the CD4 regulatory T cell, dependent on a molecule called FOXP3. There's a lot of literature for 20-some years on that. More recently, we've been interested in the CD8 Tregs, originally described in mice by Harvey Kanter, but we've done a lot of work on these to show that they are really an important, and if you don't have them, then you also get autoimmune diseases. And so we wanted to use the tonsil system to say, can we interrogate the role of these different regulatory cells, and are they the same or are they different? And this is work of Xin Chen and Mustafa Ghanizadeh, and they basically worked out a CRISPR-Cas9 deletion system whereby we could delete FOXP3 to inactivate CD4 regulatory cells or granzyme B to inactivate CD8 regulatory cells. And long story short is if you delete FOXP3 in T cells and activate those regulatory cells, now the tonsils will make autoantibodies, which they wouldn't before. You know, it involves stimulating the cells with the autoantigens, but if those regulatory cells are working, nothing will happen. But if you inactivate them, now you can get autoimmunity. So you can basically create a model of autoimmunity in healthy tonsils. And particularly it says that the FOXP3 cells are mainly geared towards regulating self-reactive B cells. We get the opposite with the CD8 cells, where we inactivate those, now we get autoreactive T cells. So it seems like they really have quite different domains of influence. There's some overlap, but there's difference in domains. And it also says that since typically in autoimmune disease you have both autoantibodies and autoreactive T cells, that you've got basically two separate checkpoints to get through. And since you can see autoantibodies years before clinical autoimmune disease, it suggests that that's the first checkpoint you have to get past. But where we've seen these KIRS CD8 T cell regulatory cells, CD8 cells, active is in infection. And we know infection typically occurs just before clinical autoimmunity, as Joe is showing in his system. So we think this is the second and most critical checkpoint before you get some real trouble and you show up in the hospital. The other things we've been really interested in is can we combine these immune organoids with other tissues from human beings and get meaningful interactions. Now, if we were bioengineers, we'd be building an apparatus with bells and whistles and complicated stuff. But we're molecular biologists, so all we do is we throw stuff into the wells and hope something good happens. And what happens here is that you put a random piece of skin in with a tonsil organoid from a different donor, you get a massive alloreaction. That is, the tonsil cells attack the skin because it's a foreign. It's like basically a transplant rejection. You get T cell and B cell infiltration, and you get killing. This is a tunnel assay showing epithelial cells being killed in the presence of a tonsil from an unrelated donor. And then we can inhibit that with cyclosporin or FK506, a classic transplant rejection drugs. So basically, we've created an in vitro model of skin graft rejection, which is kind of really old-time immunology, even before my time. But we're not, and we're carrying that over. Elsa Sola is particularly as a hepatologist interested in a liver model of transplant rejection, and we're working on that with Carlos Esquivel. But we also really, really wanted a system whereby we can get synergy between an immune organoid and a tissue in the context of an infection or a vaccine response. And it turns out this is nearly impossible to get from living people. So we had to go to people that had no choice, and these are organ donors. And it turns out that when surgeons go to harvest organs from donors, they just scoop out the whole insides, basically, with heart and kidneys and the spleen. But then when they start distributing it, no one wants to transplant a spleen. It would be, you would not want the spleen of a stranger. It would really be a bad scene. So they give it to us. So we're collecting these spleens, and it's great. It's a whole almost the immune history of a person, as well as a substrate for these immune organoids. And in fact, the spleens are even more diverse than tonsils, and they will support vaccine responses that we've had trouble doing with tonsils. And they have NK cells and other things that I know Catherine is very interested in. And we've seen, say, response to yellow fever vaccine, which we never could get a good response in tonsils, but we can get it in both antibodies and T cells in the spleen organoids, at least on a good day. And that says you can support de novo responses, probably because there are a lot more dendritic cells in spleens than in tonsils. But what we're also interested in is just human diversity. And this is an out-of-date slide. We have over 80 some spleens that we've collected. You will someday hear about the famous spleen bank of Palo Alto that we basically we'll have at least 100 spleens in which we have aliquots. So you can get up to 10 billion cells from a spleen, and you can freeze down aliquots of 100 million cells each, dozens and dozens. So you go back to the same donor over and over again, and you can look at human diversity, because that's another thing we're keenly interested in, because people are diverse. They respond differently to vaccines and everything else. So, but we don't have a way of capturing that with any great resolution. This will give us a way to do that, and also look at active responses like vaccine responses or drug responses or other kinds of things. And we're already, on the way to analyzing those things. But what I want to tell you about that's most exciting is working with Calvin Kuo, you've heard about, and this is the second generation lung organoid system, and this is a system whereby the lung organoids are more diverse than they were originally, and also that they have immune cells. And he's infected these with SARS-CoV-2, as you heard in the earlier experiment with Catherine. We actually helped characterize some spike-specific T cells that were stimulated as a result of this. But what really attracts us and fits with the organoid program is to combine lung organoids with spleen organoids. And there are a lot of technical issues with that, but this has been the work of a graduate student, Azim Mosen, who's sitting in the back there with VAMC. And so here we're basically making lung organoids and spleen organoids, and then infecting the lung organoids with flu or TB, and then looking to see if we get a response. And this is the co-culture system that Azim has developed. You can, in a gel matrix, you can actually tell which are the spleens and which are the lung organoids. One of the predictions, or what people think happens in an infection, is you get an infection, a respiratory infection in the lung, and then dendritic cells traffic to the lymph nodes and other immune organs like spleen and stimulate a response, B cells and T cells, and then those B cells and the antibodies and the T cells should then migrate to the site of infection and do battle there. So is that happening in this system? Well, first of all, we see antibody responses, anti-flu antibody responses coming from the spleen if there's an infection in the lung that's adjacent. We see increases in plasmablasts, and we also see CD8 T cells that are trafficking to the lung organoid to kill flu-specific cells. And this is a flu-specific tetramer staining those cells. And we know these came from the spleen because we labeled the spleen cells with cell tracer. So anyway, so it looks like, is this a perfect system? I don't know. We're gonna find out, but it's already got the hallmarks of the sort of communication that we would expect with a working immune system fighting an infection. And the next step is actually now to use vaccines to say, can we blunt the infection with a vaccine? And there are all kinds of tricks, and there's some early data, ASMs that suggest that that's probably going to be true. So very lastly, hopefully I have a few minutes left. Is that true? Oh, good. Okay. I was going to talk about something else, but in the context of this meeting, I think this is quite relevant, and it also picks up where Catherine left off, although he left it abruptly in terms of sex differences. So this goes to the whole business of long COVID, which of course now is part of what everybody worries about, but also that it's not just COVID. It's really, in this review by Kiko Iwasaki and her colleagues, it's something we've been seeing after severe infections. And basically, some people just seem to have things happening afterwards. MIS is just one of those things, but this is more what's happening with adults is this long COVID phenomena where you have COVID, you get over it, apparently, but you are not over it. There's fatigue, there's other stuff. And this also is, also connects with this other phenomena called ME/CFS, or chronic fatigue syndrome, which is of unknown origin, but has similar symptoms. And in fact, the clinic run by Hector Bonillo here at Stanford combines the long COVID patients with ME/CFS patients because they're so similar, and yeah. So, and here are some of the symptoms: severe fatigue, post-exercise malaise, all kinds of things. But this fatigue is a similar phenomena. So, Vishnu Shankar, a graduate student, got really excited about this disease, starting with the chronic fatigue, and you could say it was from first principles or simplistic to say, well, fatigue must have something to do with energy metabolism. And he really pursued that with a fantastic focus. And he found something. And basically what he found is that if you look at reactive oxygen levels, mostly in mitochondria, that he could see that both ME/CFS patients and chronic fatigue patients seem to have, long COVID patients had elevated levels, CFS more than LC. But what was even more interesting is if you separated males from females, you could see that the ROS elevation was very much a female aspect of this and not affecting males. But again, males are patients, so isn't there something there? Well, there is. So if you look deeper into, that was chronic fatigue, this is long COVID, you're basically seeing the same thing. Again, females with this syndrome have elevated ROS levels, males do not. But then looking deeper at other aspects of mitochondrial or oxidative damage, there we could see that both males and females had elevated glutathione levels and a whole bunch of other stuff that I'm not showing regarding damage to lipids, mitochondrial lipids. So it seems that both males and females with these syndromes have oxidative stress, mitochondrial damage, but there's quite a difference in terms of male and female in terms of ROS levels. It also turns out ROS is elevating ROS is important for lymphocyte activation, but then you got to shut it down because it's not good to have oxygen radicals in your mitochondria. That's going to mess things up. So it seems like males probably had destructive ROS early on, and then were able to shut it down, but they still retained mitochondrial damage. And so you have less ability to generate energy. Now, why would that give you chronic fatigue? Well, and why are we looking at lymphocytes? This is we should be talking to neurologists about this, right? And I asked Hector, don't you talk to neurologists? He said, no, he tried, but they didn't return his calls. They didn't want to be bothered by this. Well, there's a very simple answer, is that it's easy to get lymphocytes, but it's much harder to get brain biopsies from healthy individuals. Core samples, no, it's just not, it's not going to happen. But there's actually a rationale for getting lymphocytes because the immune system uses up a lot of energy. It uses up, it's estimated 15 to 20% of the body's energy. I mean, the brain people are always talking about how the brain uses a lot of energy and stuff. But lymphocytes are right up there, and there are a lot of them. And the other thing is, think back the last time you had a serious cold or flu. You felt like shit. Do you feel like running laps around the track? No, you were just lying there moaning, and you had no energy to do anything. So we're postulating this radical idea of an energy sink, that this is creating a lymphocyte dysfunction, and it's also it's even worse because there's hyperproliferation and things like that. So your immune system is running in circles trying to recover, but it's not recovering, but it's using up a lot of energy in not recovering, and that means you don't have any spare energy to run around the block. So this was going to end at some point, and I'll end with some conclusions, which is something we've been wrestling with human immunology for decades or ever, is how do we do mechanistic experiments? It's always been the excuse that working on inbred mice is you can do mechanistic experiments, and you can't do those in living humans. But with these organoids and the ability to do knockouts, it takes 2 weeks to do a knockout in an immune organoid. It takes a year to do a knockout in a mouse. There's some serious advantages here in terms of genetics and testing hypotheses. You saw the example of the flu vaccine. We basically were able to test the hypothesis without a human cohort. Now, does that mean it's true? Yeah, I think you'll have to do a human cohort, but still, you can't do a mouse cohort because the mice have only one class 2 MHC, the black 6 mouse. So it's off the table to be able to look at MHC class 2 polymorphism, or it would be very difficult to do. So we could just do it quickly in the organoid and get an answer that made sense and bolster the hypothesis. And I just saw that this will be coming out in Science in a couple of weeks. And that's the story with the flu strain. So I think we can move away from the original sin, as exciting as that is. It reminds me of a famous Tom Lehrer song, actually. We can talk about MHC class 2 polymorphism, and you could predict that. You could basically plug and play and say, okay, this person has these class 2 MHCs. What kind of peptides would they be making from this pathogen, whether it's flu or anything else? And then I had to detour a bit to talk to you about regulatory T cells and how it seems like we can separate CD4 regulatory cells from function of CD8 regulatory cells. I talked about spleen organoids give us a broader range of responses, and particularly with the connection with organ donors, we're able to access all kinds of other tissues besides spleen to look at those interactions that we know, and we're getting some interesting data there. And then lastly, talking about a solid link between long COVID and ME/CFS patients, and probably these other patients that are having severe secondary problems after infection, and that maybe this all can be traced to oxidative stress and mitochondrial dysfunction, and maybe and that there's this really striking difference between men and women. So we'll thank, of course, all the people that have contributed and been incredible lab members or collaborators or funders. I mentioned Vamsi, who's also in the back there, willing to accept any praise that anyone might have to spare for him. And also Azim, as I mentioned, Elsa Sola, Chen Wang did the rejection experiment with skin. And I talked about the knockout experiments at Shin Chen and Mustafa. That paper actually is also in press in Nature Immunology, and lots of other great contributions, some of which I couldn't talk about today, and great collaborators like Calvin, Catherine Blish, Ron Davis, Hector Bonilla, et cetera. So thank you. I'll stop there.
One, I think the whole chronic fatigue syndrome piece shows how our own personal blind spots resulted in us not doing the right science 40 years ago, and we would have been in a lot better place for long COVID. And I think all of us have had chronic fatigue patients in our clinic, and I think we just, for almost 2 decades, never took them seriously. And I think if we had, we probably would have had some of these early findings and probably a solution. Secondly, I just have to say, RV144 was not a failure. That was my trial. It had 50% efficacy at 1.5 years. But because we wrote it that it had to have 3 years of efficacy, we deemed it not successful. But in today's parlances, 50% efficacy at 1.5 years would have been incredibly successful and 10 times better than our current COVID vaccine.
You're moving the goalposts.
So we moved the goalposts backwards for vaccines. So I just want to say that, yes, and a third were still protected at 3.5 years. So it just shows that even though that was only a study from 15 years ago, how far we've moved backwards, both for the malaria vaccine and accepting a short duration, lower efficacy.
That is a really terrible vaccine.
And for COVID, well, we're using it. And for COVID, a very short duration of protection. So I think we have to, maybe when we're thinking about vaccines, see them almost as a prolonged therapeutic.
A glass half full.
Well, prolonged therapeutic in some cases.
And actually worthy of study. You know, could we dig in? Could we take their spleens, for example? Or tonsils. Tonsils. You know, people are doing amazing things with biopsies of lymph nodes, and I think doing biopsies of tonsils would actually be pretty straightforward. So things are doable. Yeah. But thanks for that corrective. I tend to, overstate everything.
Well, I think we're going to move on to our next speaker.
All right.
Thank you.
Sure.